Hello, I'm David Plachetzki, an associate professor in the Department of Molecular, Cellular, and Biomedical Sciences here at the University of New Hampshire. Work in my lab spans a range of different topics, from ecological genomics to ecophysiology, sensory biology, animal microbe interactions, genome innovations, and we're always interested in broadening participation whenever we can. The question that we always think about in my lab is, how do marine invertebrates evolve to interact with their environment? We study this using genomics, a range of different types of experimentation, and fieldwork. And today I'll talk just very briefly about 3 different projects that we have underway right now. The first of these is interested in understanding why fouling marine inverts settle where they do. And this is interesting to us because it turns out that they use some very simple nervous systems and sensory systems to determine where to settle and where to not settle. And this could be potentially interesting to aquaculture or other industries that are ocean-based, because these marine organisms cause a lot of problems with their fouling activities during the summer months. But we've uncovered certain sensory parameters, if you will, that they really don't like. And so it could offer a pretty easy, cost-effective, and environmentally friendly way of keeping these things off of aquaculture. And if you'd like to talk about that, please stop by the table. Another thing that we've been thinking about a lot is how marine sponges work. So we know that marine sponges are absolutely filled with microbial symbiotic communities that really serve a lot of different functions, important functions in marine ecosystems. And it turns out that when you look at the genomics that underlie these symbiotic communities and the genes that they include in their genomes, we can decipher what types of metabolic strategies the symbiotic communities are using. And this is basically sort of like a cookbook for how to put together new microbial communities to serve different tasks. And one way to think about this in terms of industry relevance could be that we could potentially build bioremediating sponges using engineering approaches that could clean up oil spills or something like that, or eliminate toxins from the water column, or even make ecosystems and reefs that are threatened right now more healthy, just by tweaking the composition of the microbial symbiotic communities. And then the final thing that I'll talk about today is actually kind of interesting, kind of strange. We're really interested in hagfish and why their slime is so slimy. So hagfish slime is really remarkable. It's really unlike anything known from other vertebrates, and it's also kind of disgusting. So we want to understand, we really want to understand how this evolved. And so to do this, we've been sequencing genomes of hagfish and slime glands from around the world, and we've uncovered a set of novel genes that underlie this obvious novel phenotype here. And so that's also interesting from the point of view of biomimicry. There could be possible textiles that could alternative biodegradable textiles that could come out of this. And so that's another way that sort of genomic prospecting is being used in the lab. And so if you're interested in discussing any of this, please stop by the table or get in touch with me. This is the QR code for the lab, and I'd be happy to talk to you. Thanks.